Okay, first of all, I would like to thank the Society for Applied Microbiology for inviting me to deliver this talk. And also, I would like to thank the Society for supporting our journals. My talk is going to center on the lifestyle of Pseudomonas putida, and particularly how these microorganisms can be used in agriculture and also for the bioproduction of chemicals. In general, the idea or message I want to convey is that the knowledge, the insight we have on the life of a microorganism can be used to develop new biotechnological processes. And this gives rise to the double style of the life of Pseudomonas, the natural life and the life that we can have through the manipulation of this microorganism or the use in biology. We all know that the life on our planet is based on the continuous cycling of the elements, carbon, nitrogen, sulfur, iron, etc. This is an oversimplification of the carbon cycle, where we have CO2 that is fixed by the plants, and this is a way to store carbon. This photosynthetic carbon enters the food chain and through respiration will return CO2 to the atmosphere. What is probably less known is that about 20% of the carbon fixed by the plant is exudated to the soil, where it serves as a carbon reservoir that can be used by microbes to thrive, and also again in the respiration they will return CO2 to the atmosphere. All the elements in the cycle are needed to get the cycle going on, but this allows me to put microbes in the center of the cycle. When I sent a summary for this talk, I mentioned that we can distinguish between two types of microorganisms: cosmopolitan and specialist. Cosmopolitan microbes can be isolated from any country in our planet, from the root of plants, or pristine lakes, cultivars, etc. While specialists are isolated from defined niches. These can be artificial niches, like for instance, wastewater treatment plants, or polluted sites with hydrocarbon, or could be isolated from natural places like hot spring or very acidic rivers like the Tinto River in Spain. Pseudomonas putida is a cosmopolitan microbe. It belongs to the genus Pseudomonas that originally the first description was done by Migula in the 19th century. The first article on microbial ecology of Pseudomonas is back to 1926, where microbes that were identified as potentially Pseudomonas were isolated all over the US from different soils as able to grow and degrade different compounds. Pseudomonas putida is a member of this genus. They became raised interest in the 60s when they were described as able to degrade a number of aromatic hydrocarbon, linear hydrocarbon, etc. In the 80s, when were declared a safe strain for genetic manipulation and degradation of xenobiotics. They are gram-negative bacteria with two membranes, and they can be found as free-living microorganisms or very often associated to abiotic and biotic surfaces producing these biofilms. Probably one of the most studied strains in the species is the one named KT2440 that derives from the NT2 strain that was isolated originally in Japan as able to grow on methicillin and toluene. The genome of this strain was the first to be sequenced in the early 2000s by a consortia of American and German group mainly. It was shown that Pseudomonas putida is about 6 megabases, and what I would like to call your attention is that they have 7 full rRNA operons. And within this operon is the 16S RNA sequence that have been used in taxonomy to organize microorganisms. Norberto Paleroni and Stanier and Dudorov in the 60s of last century, using a series of metabolic tests, established the different species within the genus Pseudomonas, and then Norberto also, with the 16S, defined what is now known as the Pseudomonas group. And I have the opportunity to discuss with Norberto a few years ago when he came to Granada about the strain we isolated. And Norberto said that we cannot use a single gene to define a genus or a species. We need to go a little bit deeper. So we need to establish the limits of the species, and to do that, what we had done is to sequence a number of microorganisms or annotate sequences that were available in the data bank, or use sequence annotated by other research groups. As I said, Pseudomonas putida is a cosmopolitan microbe, and this can be shown here. For instance, these two strains have been isolated in Spain, this one in France, originally in Japan, the US, Vietnam, in Europe, and this is not only Pseudomonas putida, but it's an endophytic microorganism that live inside the root of different plants. So what we have done to define the limits of the species was to compare all the genomes of these species and a few others that we also sequenced in Granada. And we start with comparing 2 genomes, 3 genomes, 4, etc. And here what I show you is the number of genes that they share, all of them share. So all of them have about 6 megabases and encode about 6,000 genes. And what you can see is that at the point it became asymptotic, and this is about 3,000 genes. So the limits of the Pseudomonas putida is that this microbe of this species share about 3,000 genes. This set of genes is called the core genome. And when we analyze, then we have identified a number of genes and functions. For instance, addition for attached to surfaces, a number of central metabolism, chemotaxis, catabolic pathway, for instance, for benzoate degradation, stress resistant determinant, and a number of RNA polymerase elements. So basically, that set of genes, what it's doing is defining the basic physiology of Pseudomonas putida, and also define where Pseudomonas can live. Obviously, all the strains got about 3,000 genes that are not shared by all of them. They could be shared by 2 strains or more strains, but not by all of them. Those genes are called accessory genes. And sometimes those accessory genes can give rise to the same phenotype. For instance, many strains of Pseudomonas putida will be able to degrade toluene. But Pseudomonas putida has at least 3 different catabolic pathways for degradation of toluene. And those genes, the genes in Pseudomonas, they are different. So they are not part of the core gene, but they are accessory genes. Not all strains of Pseudomonas are able to degrade toluene because in some cases, the toluene degradation genes are born on plasmid that can be moved from one species or from one strain to another. Although I'm not going to enter into the pangenome of the Pseudomonas putida, I want to say that every time we have sequenced a new isolate of Pseudomonas putida, we have found that they has between 50 and 300 genes that are unique for the species. So that means that Pseudomonas putida is a species that is in expansion. So we haven't reached yet the limit of the genome of this microorganism. What we have with all this genetic information and the experiments that have been done before is a massive amount of information that we store in databases. This information can be used to define in silico models and predict functions. And with this information, what we do is to go to the laboratory, do experiments, and then go back to the model to refine it. So in this way, we have been able to define new functions, to refine new catabolic pathways. Even we have been able to define new structural elements of Pseudomonas putida. With this information, let's say in the last 7-8 years, we have been focusing on several aspects of the biology of Pseudomonas. And the lines we are running in Granada related to gene regulation and signaling. Originally, we worked quite a lot on the toll catabolic pathways. Then we also have been working on central metabolism and other pathways. More recently, we are using Pseudomonas putida as a model system in agriculture and biotechnology. And we have been working on the formation of biofilm. I'm going to talk a little bit today about these 2 aspects of the biology of Pseudomonas. I mentioned that Pseudomonas is a cosmopolitan microbe. A number of people have been able to isolate members of this species in different countries, in different environments. We in Granada have isolated Pseudomonas putida from wastewater treatment plant and also from soil. But what we have realized is that one of the favorite niches of Pseudomonas putida is the rhizosphere of plants. So in the rhizosphere is the soil associated, closely associated to the root of plants. And as I said, 20% of the CO2 that is assimilated by the plant appear as organic carbon in the soil. There are sugars like glucose, basically the 20 proteinogenic amino acid organic acids, and other. And this makes the root, the rhizosphere, a relatively rich environment in which Pseudomonas can proliferate. But Pseudomonas not only proliferate there, but also exerts some positive effect on the plants through nutrient mobilization, production of phytohormones that help plants to grow, and even induce systemic resistance to protect the plant against pathogen. I'm going to show you some of the experiments we have done to study this plant-microbe relationship. And this experiment I'm showing you here, what we did was to take corn seed, we submerged in a suspension of Pseudomonas putida for 10, 20 minutes, half an hour, whatever. Then we washed them very well, and we took this seed to the scan electron microscopy, and the dots you see here, they are Pseudomonas putida. Between 10 to the 4 and 10 to the 5 cells of colony forming unit are attached to the seed of corn. This, if you do the same experiment with tomato, beans, or other plants, you also will see something similar. If you allow the seeds to germinate, they will produce roots, and bacteria will establish and will grow, will use the exudate, and they can reach level in the order of 10 to the 7, 10 to the 8 colony forming unit per gram of root. Let me show you what happens when Pseudomonas see a corn grain germinating. What you see here, this shadow is the corn, okay? And what we have done is just we place Pseudomonas at a certain distance from the corn. What you see, Pseudomonas start to move, and the cells are moving towards the seed. That is chemoattraction. We have found that Pseudomonas got a number of chemosensing elements on the surface that are able to detect amino acids, Krebs cycle intermediates, and all these compounds are exudated by the bacteria. What you see also here, the cells are moving, but when they reach the surface, some of them start to stop. They will start to produce some microcolonies, like the one over there, and start to grow. This is a movie taken a little bit later. What you see here, for instance, they are just producing microcolonies. They start to colonize the full surface, and when they are very close, they stop moving. What you have seen there is summarized in this overhead. What we have is bacteria. We got plant signal. The bacteria use the flagella, obviously, to move and to attach. At the point they will bind to the surface, and after a while, other bacteria will be attaching to those that already attached to the surface. We know that the bacteria in those microcolonies can grow and reproduce, and eventually they will be released from the biofilm, responding to new signals. So the question is, which genes are important in these processes? And we have taken different approaches to identify those genes. One of them has been to create a collection of mutants. We use mini-10-5 mutagenesis. That was a method developed by Victor De Lorenzo and Ken Timis in Braunschweig. I will not go into the detail, but we got the mini-transposon inserted in the chromosome of the wild-type strain. We array those clones. We use southern blot to confirm that the insertions were different, and later on, Estrella sequenced about 5,000, the location of about 5,000 independent genes, and we got more than 3,000 different genes knocked out in the genome of Pseudomonas. So we have a wide collection of mutants to analyze. With this collection of mutants, we had used robots to analyze different functions. And once we identify function, we have been also through transcriptomic studies. And to make a long story short, we have identified a number of genes that are related or required for addition to surfaces, for instance, flagella, type 4 Phylli, addition protein LAP-A and LAP-F, exopolysaccharides. We also have identified genes that are required to survive in the root of the plants. For instance, a gene related to respiration, iron sideroff, ROS, reactive oxygen species, a protein that are related to removal of those species, and efflux pump that pump out a toxic compound from the cells to the outer medium. I'm going to, although we have characterized many of those mutants, I'm going to talk a little bit about two of the mutants we have isolated. Those are LAP-A and LAP-F. The reason is that they are core genes in Pseudomonas putida, but also they are the longest gene in the species, and probably one of the longest proteins in the prokaryotic world. LAP-A is about 9,000 amino acids. LAP-F is above 6,000 amino acids. They are organized in domains with different size or repeat of amino acid, and they have also a calcium binding domain that is critical for biological activity. These proteins are related to adhesion, and as such, we were expecting that they would be located on the outer surface of the outer membrane. And that is the case with LAP-A and LAP-F. This, for instance, is a series of experiments done in George O'Toole laboratory with antibody against LAP-F and immunostaining. You can see the protein over there. And we now, through other series of experiments, know that they are a specific system to transport this protein to the outer surface. Let's see how the role they play, okay? And to analyze this, what we did was to label the cells with green fluorescent protein and follow the formation of biofilm. So indeed, we know that the first element to touch a surface is the flagella, and then the bacteria, let's say, turn and fix. And we, after 2 hours, 4 hours, 6, 24 hours, you can see that the wild type is able to produce a thick biofilm. When you do the same experiment with the LAP-A mutant, what we found is just, well, the biofilm didn't develop, and just we saw very few cells attached to the surface. With lab F, we are in an intermediate situation. We see they fix, but they are not able to develop a sick biofilm. This property or this ability to, or this defect in binding to surfaces confer negative advantage to the mutants. If you compete wild type and the mutant, what we see in this case, we label the wild type with the N cherry, so they are red fluorescent. Lab F, in this case, green fluorescent. We start experiments with the same cell density, the same number of cells, and we monitor the population with time. You see that the wild type will dominate always. The longer the time, the higher the domination. And even if you look under the confocal microscopy, you see that the red side, the red cells dominate over the green one. So what we can see with biotic and abiotic surfaces is something very, very similar. Bacteria reach the surface, they bind to the surface, and this orange stick you have there, that is lab A, that is the protein that is used to stick to the surface. And the green one is the lab F. So a mutant in lab F is able to produce this, is able to bind to the surface, but is not able to develop these microcolonies. So that is why it's not able to produce a full biofilm. Given that Pseudomonas putida lives in the rhizophyll plant that is responding to plant signal and is able to develop in the root, we have studied this species as a model system in agriculture. And this overhead summarizes a number of the properties we have found. Pseudomonas putida is a PGPR protein, a microorganism, is a plant growth promoting rhizobacteria. It's able to solubilize nutrients, for instance, phosphorus, and this can be done through two different mechanisms. One is the production of organic acid that solubilize phosphate at the local level. They also produce phosphatases that release phosphorus from organophosphorate compounds. It also allows mobilization of nitrogen through proteases and so on. It produces phytohormones that will allow plant to grow, and I will show you one overhead later that favor plant growth. Pseudomonas is able to produce a molecule called siderophore. This molecule binds iron. Iron is very abundant, but extremely insoluble. So this siderophore binds iron, and the bacteria taking up it to satisfy its own iron need. But most important, or very important, is that this siderophore bioburden with iron can be taken up by the plants to satisfy their own iron need. Iron cycle in the soil is very important because iron at high concentration is required by pathogenic fungi to grow and to produce certain diseases. So binding or sequestrating iron is a way to avoid the proliferation or a way to reduce the proliferation of microorganisms. Pseudomonas putida use a system called the type C secretion system. That is a system that has been recently described by a former PhD student of mine here at Imperial, in which this spike enters into a number of host cells and kills them. So Pseudomonas is able to control the proliferation of certain microorganisms. And also, we have found that Pseudomonas putida is able to produce a number of volatile molecules that result in what is called induced systemic resistance. This is molecules that are produced in the root of the plant, start a signaling system that makes that the plant defends against the attack by pathogen in the aerial part. And this is something I show you here when this is a provoked infection by Pseudomonas syringae. If you don't have Pseudomonas putida in the root system, 100% of the plants get infected. But if you have Pseudomonas putida, according to the density, you will prevent the infection. So Pseudomonas putida is doing quite a lot of things that help the bacteria to grow. Some of the effects we have seen, for instance, is that Pseudomonas putida stimulate the production of secondary roots. In this case, it's corn, but also with other plants. This is important because this secondary root is the main way to take up nutrients, and as a consequence of an increase in nutrient uptake at the early stage, you can see significant differences in the growth of plant. This is tomato or green pepper and so on. So, but not only that. It's not only that the plants grow at the beginning a little bit faster. At the end of the day, they reach more or less the same height. But the quality of the fruits also increases. And with strawberry, we have seen that the yield can increase around 10%. And the quality of the fruit in strawberry, in Spain, there are 4 qualities for strawberry. The ones that are best paid are the first quality. So there is an increase in about 15% in the first quality strawberry produced by the plants. As I said, these effects are multifactorial. There are different elements, different genes involved in all of these effects. But one of them is that Pseudomonas putida is a natural overproducer of indole acetic acid. That is a phytormone. And Pseudomonas produce this compound through 2 parallel pathways. We have mutant in this pathway, or in this pathway, or in the 2 pathways together. And when you reduce the amount of indole acetic acid, the PGPR effects that I showed you before diminish. Okay, they don't disappear, but diminish. I mentioned that Pseudomonas raised quite a lot of attention last century because they were shown to degrade a number of aromatic compounds like toluene, xylene, and other compounds. And now I show you that Pseudomonas is able to colonize the root of plants. So at the point we decide that we could combine the ability of Pseudomonas to colonize the root of plants with its biodegradative property so that we could combat pollution. And this is what is called phytorhizoremediation. It's the removal of pollutants using plants and using rhizopheric bacteria able to degrade pollutants. We have carried out a series of essays. In one of them, we were collaborating with a Spanish company called Unión Española de Explosivos. And this company, on one hand, they produce explosives for different use, but they also dismantle obsolete munition or obsolete explosive. So what we did with them was to look for microorganisms able to degrade TNT. And we found this bacteria that we call clone A, the A from Spanish, amarillo. So in English will be clone Y, yellow, but is how we call clone A. And this is produce very nice colonies. Okay? This microorganism is a very efficient TNT degrader. This is the TNT that is solubility level in the reactor, and you see that it will decrease with time. Indeed, what we have is a transitory accumulation of nitrite. Then nitrite is reduced to ammonia and incorporated into glutamate. And from glutamate, it goes to the different amino acids, and we get growth. What I showed you here is a derivative of clone A colonizing a corn grain. In this case, what we have done is to label Pseudomonas with the lax gene, so that the bacteria is luminescent, is emitting light continuously. So we did the same. We submerged the corn in this suspension of the bacteria. And this is a picture taken with a high sensitivity camera. So what the light you see here is the bacteria on the core. Over here you see some gray hair. Those are the roots. And what I want to call your attention is that the bacteria are moving through the root. So we thought that an easy way to put bacteria in the environment to combat pollutants is just to attach to seed, allow them to germinate in the site and spread in the soil. And this is what I showed you here. This is a plot in the factory polluted with TNT. The red, the different color refer to the different concentration of TNT in the soil. For instance, this is about 400 milligrams per kilogram of soil. The yellow is 100, and the deep green is less than 20. Okay, and what we have here is the soil polluted with TNT. So what we want to do is just to put plants, and we starting in that point with the seed, but the roots will be spreading the bacteria all over the soil, and that will be an easy way to reach the pollutant. So we did that. We inoculate seed with clone A, ran for 60 days, and after 60 days, we took sample again. Okay, what you can see that we don't have any more red area. We still have some yellow area that were not very well removed TNT. But what is important is that the extension of the deep green area, less than 20 milligrams of TNT per kilogram of soil, were extending. So that is an efficient and easy way to introduce bacteria to combat pollutants. We have done some other experiments. Mediterranean countries, and particularly Spain, are suffering of forest fire, particularly in the summertime. And the result of a forest fire when you go there, it's first desolation, okay? It's a desolating environment. When you overcome a little bit the desolation, what you see is a black soil and carbonized plants. If you analyze that, what we have found is a number of monoaromatic compounds and polyaromatic compounds. And the concentration of these compounds varies with the temperature of the fire. The higher the temperature, the higher the proportion of the high molecular weight aromatic hydrocarbons. So what we have is a collection of Pseudomonas putida and other microorganisms that are able to degrade some of these compounds. We ran an experiment, a field test near Malaga, in the Montes de Malaga. This zone was on fire. The temperature that reached the fire was relatively low, only 400 degrees. Some of the fire can reach over 1500. And this soil was polluted with monoaromatic and low molecular weight aromatic hydrocarbons. So we ran a number of assays there. The control is, we use autochthonous seed, herbaceous seeds, this parcel. When we have the PGPR bacteria plus the hydrocarbon degrading bacteria, the recovery was much faster. And after a year, there was the appearance of this zone. So, and what is more important, more than 90% of the original aromatic hydrocarbons that were present in the soil completely disappear. So that, again, is a way to restore a natural environments in a relatively easy and inexpensive way. I'm going to talk a little bit about Pseudomonas putida as a chassis for the production of chemicals. And this is within the context of circular economy. The general concept of circular economy is that every item of waste that we produce should be recycled to try to produce some new compounds. This is a way to avoid pollution, so we avoid to put waste in the environment. This is a way of saving natural resources that we need for many other things. And it's also a way, or it's being a way to develop a new concept that is the bioeconomy. So it's not only that we prevent pollution, it's not only that we save natural resources, it's also a way to enter in the economic market. The idea behind this is the concept of self-factory, and what we are trying is to replace petrochemical factories by biological factories. But to replace these factories, we need to produce chemical in large amounts. So we usually, or in many cases, we are not able to scale up the processes. But this is completely possible. In fact, we should take into account that more than 99% of the ethanol used in the world is of biological origin. It's produced through alcoholic fermentation. And this has been done for years using grain or sugar cane. They produce glucose and it's fermented to ethanol. The main use of ethanol, around 70% of the total ethanol produced in the world, is used to blend with gasoline. At the end of last century, it was a big fight between the use of this grain for fuel. So the food versus fuel fight. So a number of research groups and companies start to develop or to look for ways to produce sugars from other sources. And we have concentrated in the use of agricultural ways, like corn stubble, wheat straw, bagasse from the sugarcane, woody biomass, to produce sugars and to ferment them to ethanol or other compounds. The ethanol that is produced from these agricultural waste is called second generation ethanol, to differentiate from the first generation ethanol. But ethanol is a very volatile compound, and not only from the chemical point of view, but also from the economic point of view. The price of ethanol is influenced by the price of the grain, is influenced by the price of the petrol. So this new bioeconomy, what it's looking for is ways to produce other chemical with add value that can replace chemical processes. And we have been concentrated in production of butanol. Now we are interested in production of octanol. I will show you that we can produce compounds like styrene or other compounds that could be used in the cosmetic company. Why to use Pseudomonas putida? Okay, ethanol fermentation is obviously done by yeast. E. coli is some excellent microorganism to do many things, and we know quite a lot. But why Pseudomonas putida? And the reason for that came from the fact that certain Pseudomonas putida has a number of what is called efflux pump. And these efflux pump are able to pump out toxic compound from inside the cell to outside the cell. And some of those chemicals are, for instance, aromatic compounds that are extremely toxic for many microorganisms. And then Pseudomonas putida is a unique microorganism for the production of aromatic compounds. And the reason is that these pumps remove all these toxic compounds to the outer medium. The problem we have is that Pseudomonas, as many other microorganisms, doesn't know how to produce this chemical we want to produce. And the reason for that is that the genetic information they have doesn't encode those functions. So what we need to do is to construct new pathways. And for this construction, I'm using the Lego system. With the Lego, what we have is a number of pieces, different color, different sizes. And what we do is we collect them and we organize. And we can produce a truck, boat, or can make very complex structure like a city with cars, trees, and everything, okay? Our Lego pieces are the genes. We have thousands of genes in the data bank. But we need something else in addition to the genes. We need systems that control the expression of the gene. So combining in a judicious way these pieces, the gene that we have in Pseudomonas plus gene that may come from different microorganisms, we will be able to produce different compounds. So with that in mind, we started a program to produce aromatic compounds. We concentrated on two of them, 2-phenylethanol and trans-cinnamic acid, and now also styrene. We manipulate Pseudomonas so that it can use glucose, that it does in the natural way, but also we introduce gene for xylose and arabinose degradation. And the reason for this is that with the agricultural waste, 70% of the sugar is glucose, about 25% xylose, and 3–4% is arabinose. So we need to ferment or to take up all these compounds to produce these compounds. So one of the compounds we concentrate is phenylalanine. First thing we need to do was to generate a phenylalanine overproducing strain. And for that, we block 4-phenylalanine degradation pathway. But this was not enough to overproduce phenylalanine. We also need to overexpress phenylalanine biosynthetic genes. And at the end of the day, we got a strain that overproduced phenylalanine. So that was one of our strains. So with a single gene from a cyanobacteria, we can go from phenylalanine to transinamic acid. This enzyme is phenylalanine ammonia liase. Now with another decarboxylase gene, we can go to styrene. To produce 2-phenylethanol, we clone a decarboxylase from Rhodococcus. And in addition, to get high production, we overexpress a natural gene in Pseudomonas that is an alcohol dehydrogenase. So in this way, we can produce compound that can be used in the cosmetic. The reason for that is that the smell to phenylethanol smell to roses, so that is why it's used in cosmetic, or we can use to produce polymers and so on. Another compound of interest is tyrosine. And from tyrosine, with a single enzyme, you can produce phenol, dopamine, tyramine, 4-hydroxyphenylpyruvate. We carry out a techno-economic analysis of all the potential intermediate in Pseudomonas that could be used to produce a compound of interest. And there are about 7–8 compounds that will allow you to produce economically this compound satisfactorily. But what I showed you here with tyrosine or phenylalanine, we have a compound, and we have 1, 2, 3 different enzymes, and we have 2, 3 different compounds. Okay, so we have a limited number of compounds. So something more complex is the production of a platform that will produce different intermediates from which can be produced a wide variety of compounds. And this concept was developed by Delo Monaco in the US a few years ago, when they showed that it's possible to reverse the beta oxidation cycle in E. coli, in which you start with 2 molecules of acetyl-CoA, you condensate, and through a number of reduction and hydrolysis, you can produce a number of intermediates. These intermediates with different enzymes can give rise to a wide variety of compounds. And what is interesting here is that in every turn of the cycle, you add 2 compounds. So the easiest compound is to produce N-butanol from this point. But if you don't take N-butanol and you go to one more cycle, then you will have a C4-acyl-CoA that with the C2-acyl-CoA at the end of the day will generate a C6, and then you can produce hexanol. And if you go to another one more cycle, you can produce octanol. So it's possible to produce this compound in yeast. And now we are working in Pseudomonas, mainly concentrated in the production of octanol. But you can go to different compounds as it can be required. As a summary of my talk, 4 points. The pangenomic analysis can help to define the limits of a given species. Within the pangenome, the core genes are those that define the basic physiology of the microorganism and the basic places where this microorganism can live. Pseudomonas putida is a plant growth promoting rhizobacteria. It establishes a commensal relationship with the plants. The plants feed the bacteria, and the bacteria promote the growth of the plant through nutrient mobilization, avoiding colonization by pathogen, and even producing compounds that will avoid the attack of this pathogen. If we combine the natural properties of Pseudomonas to degrade pollutants and the property to colonize the root of plants, we can develop efficient process to remove pollutant from soil in a process called phytorhizoremediation. And also because some of the Pseudomonas putida has accessory genes that allow them to be more resistant to toxic compounds than any other microorganism in the world so far. They can be used as a valuable chassis to construct a new pathway for the production of good. The work I have shown have been supported by national or regional grants, EU project. Very important. Thank you very much for your attention.